I'm Sean Ainsworth, CEO of Immusoft, where we are really excited to be developing B cells as a modality for therapeutic protein delivery. The B cell is a cell type that has two facets that really compel us to work in this space. The first being that these B cells, or an end-stage differentiated B cell known as a plasma cell, is an extraordinary biofactory. So any given plasma cell can produce and release thousands of antibodies per second into the bloodstream, and we harness that ability of the B cell to produce our therapeutic proteins of choice. The second really compelling aspect of this cell type is the fact that they will naturally engraft in the bone marrow. And so what that means is there's not a need for a preconditioning regimen as we see with some of the other cell types. Those preconditioning regimens are typically very harsh, best avoided entirely if possible, and certainly not something that you would want to redose. And so with our approach, we do believe that we'll be able to go back and redose these patients if needed or as needed. And that's also in contrast to if we think about systemic delivery of virus vectors. So we've been in this space for a while now. In the process, we've developed a pretty substantial intellectual property position around things like the differentiation protocols, our culture system, some of the compositions of matter, et cetera. Really great IP position. The other thing is our large market potential. So we started out by delivering antibodies, kind of a natural choice for plasma cells with our B cells, and then began to inquire as to whether we could deliver other protein types. And we found that indeed, given the right conditions, we were able to do that in high quantities. And so we've been delivering now antibodies, signaling proteins, enzymes, and so forth. And if we think about just antibodies and enzymes, there are just tremendous potential in those two therapeutic areas. We're really excited to be first into the clinic with an engineered B cell. Our lead program is deploying an enzyme. It's iduronidase to treat MPS1. So this is a lysosomal storage disease. There is currently a treatment on the market, but still pretty substantial unmet need. And we're hopeful that we'll be able to really show some improvements in the lives of these patients. We've done all this on about roughly $50 million in paid-in capital, so we've been pretty resourceful. We've done also, we've secured also some non-dilutive funding in the way of partnering and grant financing. And so, we think we've done quite a lot with the resources at hand. I attribute that in no small part to really a phenomenal management team that I'm tremendously proud to work with. So together, without going into everyone's background, we've had successes in small biotechs to big pharmas, taking programs into the clinic through to approval, taking companies through to acquisition and IPO. So just really a great group to be working with, and a scientific advisory board that's really world-class. So the B cell, as mentioned, that end-stage differentiated plasma cell is really just designed to produce proteins in mass quantities. And so once those B cells are differentiated into the plasma cell, they do engraft in the bone marrow, and at that point they are by definition a long-lived plasma cell. And so what we're looking at on the right here is the response, the antibody levels after various vaccines that have been given to these patients. And what we see is that these antibodies do persist out pretty stably for many years, potentially decades. And so when we look at our approach in the overall landscape, on the left here, we see the in vivo modalities of gene therapy, in particular virus is kind of the standard at this moment. Ex vivo using myeloablation regimens for engraftment of the B cells. What we have, I think, that addresses the unmet need here is the ability to go back and redose in particular, but also to dose initially without some of the toxic regimens associated with these approaches. And so on that third vertical is enzyme replacement therapy, not a advanced therapy like the gene and cell therapy approaches, but what that is really a modality that is delivering a therapeutic protein and one that we think still has pretty substantial unmet need that we can address with our B-cell approach. And so most of these ERTs do have pretty short half-lives. They require frequent dosing, and if we can ameliorate that alone, that's a huge quality of life improvement for these patients. But we also anticipate that by mitigating that sawtooth effect associated with the enzyme replacement therapies, will ultimately deliver better outcomes to these patients. Pardon me. And so from an IP standpoint, we got our start with some intellectual property from David Baltimore's lab, Nobel laureate, of course, out of Caltech. We've built on that with some of the intellectual property around the Sleeping Beauty transposon system, as well as then some of our intellectual property that comes in-house, and some licenses from third parties. So collectively, we've got globally 60+ issued and/or pending patents. We've got freedom to operate. That's been assessed by a couple of different venture funds that have come in independently. So great IP position. And so the way this works, we start out by collecting the patient's B cells from the apheresis product. Our initial approach is autologous. We program those B cells using a transposon system, so it's a non-viral approach. Ex vivo, then, we expand those and we differentiate them into the plasma cell, towards the plasma cell, maybe more accurately. And that's what our drug product is. So we administer that to the patient. We do expect to see engraftment and sustained therapeutic protein production for many years, potentially decades. And so we're starting out, as mentioned, with MPS I. The table on the left here is a number of approved protein therapeutics for treatment of various lysosomal storage diseases. And what that really represents to us is a lot of opportunity for a B-cell approach. So to be able to go in, well, let me take a step back, pardon me. In all of these cases, these are delivered by infusion, usually on a weekly basis. And the reason for that is that there's a very short half-life of these proteins. So in each case, they're also approved by the FDA, and so that technology risk is essentially removed from the equation. So each of them is individually compelling. Collectively, they represent north of a $5 billion market. And then what we're seeing on the right here is some of the directions we plan to move into as we generate more and more data, particularly clinical data with our lead program. So I want to take a moment to say thanks to CIRM, who have been tremendous for us, not just in the way of capital, but intellectual capital in support of both our lead program for MPS I and our follow-on program for MPS II. They've been great to work with. We plan to move from there into some additional genetically defined orphan indications, and then some of these larger market indications where we've actually got active programs underway. A lot of interest already from some of the big pharmas and potentially partnering there. So some great things going on in the labs. Our lead program, MPS I, is characterized by the loss of or dysfunction of the iduronidase enzyme. That enzyme is involved in the breakdown of glycosaminoglycans, and if those are not broken down properly, they build up in the lysosomes. They're then harmful at the cellular level through to the tissues, organ systems. It's a devastating disease, and these patients, if not treated, the Hurler patients generally don't live through childhood. Thankfully, there are treatments for this approach in the way of enzyme replacement therapy, usually for the less severe patients, or stem cell transplant, which is for the more severe patients. So a quick look at what we've been working on then. We see what we've got here is the result of a 6-month study in a mouse model, pardon me. So on the far left, that flat line is the enzyme level in a diseased animal. The red is 3 months post-treatment with our ISP-001 program. And then blue is 6 months post-treatment, and black is the non-diseased animal model. So what we're seeing at 6 months, just to focus in, we've got supernormal levels of expression in the kidney, spleen, liver, and lung. A little bit of expression in the heart and the brain. What we're really excited about is, despite not having tremendous expression in the heart, we still do. Whoops, got ahead of myself. We still do see reductions in glycosaminoglycans in the heart. And so that's what we're looking at here. If we look at the liver in particular, we see complete reduction of glycosaminoglycans, reduction across all of the tissue types, including the brain at 3 months. So there's really a lot to be excited about as we move into our clinical program. And so in our MPS I studies with ISP-001, we've shown really over and over again that we can reproducibly manufacture the cells. They produce our protein of choice. We can measure then that protein in the plasma, and we get a measurable therapeutic effect. We've also seen that with our ISP-002 program for MPS II. We've seen that with our follistatin program, and that's directed towards muscle wasting. And collectively, what we believe is that this really supports the strength of the plasma cell approach as a platform. And so as we move into the clinic, we're starting out with a couple of adult patients. We, of course, are initially looking at safety as our primary endpoint here. But we do expect to see biomarkers of activity in our clinical study. Those are defined as reductions in glycosaminoglycans, or increases in the enzyme in the plasma. Our study is at University of Minnesota. Paul Orchard is our PI. Paul is just a tremendous thought leader in the MPS I space. I think independently he has bone marrow transplanted more than any of the more of these patients than anyone else on the planet. And so as our timeline goes, we're really excited to be in the clinic enrolling patients, and we will plan to talk further about the results of that early in 2024. Our MPS II program, we're continuing with our IND-enabling studies and anticipating moving that into the clinic, if not late next year, then early 2025. And so, to summarize, we're really excited to be the first B-cell program into the clinic. We've enrolled our first patient. We expect to be coming out with data on that here in the not-too-distant future. Really, a tremendous platform, and if anyone is interested in learning more, I will be around for the rest of the afternoon. So, happy to take questions if there are any. All right. Thanks very much.